And I'll just go back a couple of slides if that's okay. Okay, so in our previous session on bioinformatics on infectious diseases, we talked about how next generation sequencing can yield a variety of different types of data. Different types of data can answer specific questions about differences at the nucleotide level, like we see here. In some cases, when we have access to raw FASTQ files, we can ask additional questions that can identify differential between samples. How are they different globally? Or what specific positions are highly abundant in differential mutations? We can then use this analysis or this information to structure data in terms of how it is related to each other. And we learned about associating those differences with specific characteristics of disease. For example, disease spread geographically, disease symptom severity, or specific types of manifestations of that disease at the biological level. What helps us study these types of relationships are databases of different types of data. Data on the genomic level, data on the structure, about the structures of different significant proteins. And as we have access to these different data sets and we can analyze them appropriately, we can actually find important answers to questions about origins, transmission, and the relationship between different hosts and viral genomes in those hosts. Now, we looked at, last time we looked at how specific details about those variations and mutations can really help us understand the dynamic nature of viruses when they are inside different environments, their ability to evade immune response, produce different types of proteins and protein complexes, and really link the natural biology of viral populations or quasi-species with how the disease evolves and develops in their hosts. So we also talked about how understanding of those protein sequences and structures and their function helps us also address the issue of vaccine design and ultimately understand how more severe disease is caused by some opportunistic and at the same time strongly pronounced differences in more pathogenic viruses. So today, we want to talk briefly about host pathogen interaction, right? We talk about the expression of genes in terms of gene expression or transcriptomic data that allows us to study how the host can adapt and try to overcome viral infection and interfere with replication. So here we had an overview of the typical steps that we need to process RNA-seq data. So you already probably saw this in transcriptomics courses, transcriptomics 1, 2, and 3. I noticed today, thank you, Roland, for mentioning that transcriptomics 2 is not available. I will make sure to add it so that all of you can have access. Ultimately, to process those individual reads and understand levels of gene expression, we need to first pre-process the data and make sure that we eliminate technical variation at the read level. Then those reads need to be mapped onto the reference genome, in this case, the host reference genome. And then we can quantify levels of expression. So in transcriptomics 1, there's an overview of a number of different types of pipelines that you can put together and get a table of expression. That table of expression will have samples and genes. So the samples are characterized by expressed genes. It's important to remember that the expression level can be in a number of different formats, FPKM, TPM, individual read counts, and we can normalize that data to make sure that we are comparing apples to apples. We really have replicates and groups. We really have comparable measurements in each one of our samples. So those types of pipelines are located under the transcriptomic processing pipelines, but we also have a number of additional utilities that we can use, for example, to study differential gene expression, right? When we talked about those project ideas, many of those project ideas required an understanding of differentially expressed genes. And differentially expressed genes could be visualized by looking at heat maps or volcano plots. So what exactly are those outputs? When we talk about differential gene expression, we're talking about methods like DE-seq and EdgeR. These are libraries of code in R libraries that essentially take a look at a number of samples per group, which represents a condition. And that condition can either be different from the normal, or if we're comparing two different conditions, then how are they different? But maybe overall, they're not that different, right? So for example, if you take two living lung cells, one is infected, one is not infected, a part of that cell's process is to fight off the infection. But the other part, and maybe the major part, is just to stay alive. So all the normal functionality of the cell is still there, right? And so that for us is non-interesting part of the gene expression data. So that data really represents noise because even if it is differentially expressed, it shouldn't be. But then there are some highly differentially expressed genes that represent the difference between this condition. So we can find those significant differentially expressed genes by looking at this p-adjusted value, which demonstrates that there is a statistical significance in difference of expression, and log fold change, which is a measurement of by how much are the gene expression patterns different between the two groups. When we visualize that data, you can see here two examples of the same differential expression result. And the difference are in these genes, right? Here we have the conditions or samples, and here we have the genes. And so when we visualize that data, typically it just shows us expression levels. But if we only put in differentially expressed genes, it will show us a clear difference between those conditions. Now, sometimes these conditions have complex patterns, and it's not enough to just look at one versus the other, one group versus the other. So we also discussed how additional data sets or additional variation in those data sets could be discovered using tools like factor regression analysis or even different machine learning tools. So those tools are all described in Transcriptomics 3. Now, since some of you are thinking about these gene expression data analysis methods and analysis of those types of data sets, I think Transcriptomics 2 and 3 are going to be beneficial. We still have time to complete those if you are interested in. So I'm going to add all of those also to the program. You can take a look at them. Transcriptomics 2 will talk about differential gene expression and factor regression analysis. And Transcriptomics 3 will talk about clustering and classification, methods that help us understand groups based on expression data and find differences between those groups or train a model to predict what those differences might show us in another data set. So when we think of gene expression response to a virus, for example, a virus that has a specific biology and that biology interacts with the host, right? You remember probably this scheme. Here we talked about how one of the options to study this variation is by looking at high throughput sequencing. So before we looked at amino acid sequences, and we saw how analysis of differences in amino acid or nucleotide sequences can help reveal global, local, or specific type of comparative to other sequences, right? We looked at 3D structures and identified specific pockets or specific regions that might be of interest for a specific condition or variation. But when we talk about high throughput data, we can look at gene expression data and try to understand how this gene expression data can help us understand the function of a protein in a specific host, right? So last time we talked about these complex protein machineries that interact with the host at the level of a standard process that is designed to identify these opportunistic viruses and interfere with them. And those are essentially these cytokines that go and try to interfere. So the virus can use some of its proteins and complexes of proteins to interfere with those standard processes. So when we look at this kind of data, right, we need to design the analysis appropriately. We need to have a condition that represents infection, a condition that represents control, for example, mock infection, and we can look at variation, for example, something with a protein mutation or protein variant that we want to understand the function of. And here in this study, there is a delta ORF6, a genomic sequence without the ORF6 gene. And here you can see the analysis done with differential gene expression and then pathway regression or pathway analysis right here, right? So these types of studies really allow us to find global differences in gene expression and link them to specific variation that we can talk about in terms of coronaviruses like we saw before, or you can see here evasion of interferon responses by Ebola and Marburg viruses, right? So what we talked about in the beginning of the program. Many of these data sets are available on NCBI, and in fact, more and more of these data sets are going to be useful for what's called network analysis and high throughput screening. So a lot of these are going to be designed around understanding interaction between different genes and different proteins, but also we can apply it to the study of viral, antiviral drugs that interfere with viral function and vaccines that are designed to train the immune system to detect these viruses and prevent their infection manifesting in the organism. So these paradigms actually are important in the context of infectious diseases to be able to study protein function and to be able to screen for effective strategies to interfere with the protein function as the virus starts replicating. So finally, about this independent project, I think it's very important for us to find a common kind of goal for this program. And that could either be that you find a project that somebody else is working on and join their project, or it could be even developing a project common to the whole group. So for example, what I think would be very interesting for us to try and accomplish is contribute to the investigation of the origins and pathogens of this or pathogenesis of the novel coronavirus, right? If we gain a deeper understanding of the coronavirus, we have great examples to follow, and even repeating that kind of analysis that we reviewed in this program will be sufficient, being able to explain what data sets you chose, how you performed the analysis, and then how you actually identify a specific feature. And maybe you want to focus on some aspect of visualization or some aspect of biological interpretation, right? That is still a great project to undertake as an independent project. And then of course we have these prepared data sets that we can also make available to you if you're interested, that require you to follow the specific steps of analysis described in the project. And so if any one of these actually looks interesting to you, I'm happy to start sharing with you more information on how you can get involved in those projects. So this session is bringing us very close to the end of this program. This program will end by the end of this week. Like I mentioned, I will make those additional courses available and some of these projects as well. You can take a look at some of the other projects that others have submitted, or explore some of these data sets that I will share with you. And then whatever you decide to keep working on, you'll have some additional time independently that you can explore the data sets and come to a presentation that you can share with me. So at that point, when you have a presentation, you have some analysis that you've done, and you want some feedback, we will actually try to make sure that the analysis is rigorous, your conclusions are solid, and hopefully some of you will be able to take the projects further in your own research. So again, thank you for joining today. will pause the recording here. And if you have any questions about finding the data sets or specific types of analysis that we discussed, or even the data sets that I'm showing right here on the screen, I'd be happy to answer those. Okay, so did anyone have any other questions about these?
No, sir.
Okay, great. So it's great to see you again this time. I will look forward to seeing some more project submissions and refinement, right? If you want to just refine your project submission, you go back into that form and you can change it and submit it again. We will see the notification by email. If you have any questions, post them on the forum, and I will see you again next time.
Thank you.